I'm Jonah Comstock, editor-in-chief at pharmaforum. I'm here at the JP Morgan Healthcare Conference in San Francisco. I'm at the Zeppelin Hotel with Dr. Adi Haas, Chief Executive Officer at AffaMed. Thanks for meeting with me this morning. It's early. I've been time-stamping some of these. This is our first meeting of the day. How are you doing?
Thank you. Good morning. I'm doing well. So it's the third day of this important conference. As any year, we missed a little bit the interactions that we couldn't do because of COVID. So being back and having live interactions like these interviews is obviously a great opportunity. So everything is good. Thank you.
So tell me a little bit about AffaMed. What's your guys' main focus, and then what are you focused on here this week? What brings you here?
So what does AffaMed do is, let me start with the vision. We work in cancer, and when you read our vision, what's interesting is we talk about derailing of patients' lives. So we see cancer as a disease that is not just a personal disease, it's a disease within families. And we're looking to address this with novel therapies. And the idea that we have been exploring now in the past 10 years is that we're looking for activation of the so-called innate immune system. An activation of innate immune system is required in order to generate a general immune response. So if we could basically activate the innate immune system within the cancer patient, you can see long-term responses. That's the idea. The reason is that you're not just generating and activating natural killer cells and macrophages, but you would also get a T-cell response. So how do we do this? We have developed a platform which allows us to generate bispecific antibodies that can, on the one side, dock to cancer cells, so any cancer antigen, and on the other side, they can dock and bind very strongly to natural killer cells and macrophages. So you see thereby you're basically redirecting initially the patient's own immune system to attack the tumor. And what we have now seen as an evidence, we're also getting an uptake of T cells into the tumor. So the first step is achieved, that we've seen not only an innate immune response, and the next steps are now that we're optimizing now the efficacy within these patients.
And where are you along your journey? How many products do you have? What phase are you in?
So we have 3 clinical programs. The most advanced is a hematological program called AFM13. The target antigen is CD30, which is expressed in Hodgkin lymphoma and in T-cell lymphoma. It's a validated target as a product is on the market, but this product is an antibody drug conjugate. Quite successful, but it acts more like a chemo. And here I come back to what I said as a vision. What we want to do is we want to give patients back not only a therapy that works, but also has a pretty good safety profile. And that's what we have learned by treating patients with AFM13. It's a very benign safety profile, so patients can come in and walk home without any side effects. We also have seen with a single agent good responses in Hodgkin lymphoma, but also in T-cell lymphoma. What we were doing next was quite interesting. As I said in the beginning, we were looking to enhance the initial responses and have learned that we can do this by simply replacing patients' own NK cells, natural killer cells, by new natural killer cells that come from healthy donors.
Interesting.
And that's a novel approach that we're now pursuing since about 2 years and have learned that this can achieve efficacies in patients above 90%. So very sick patients that have undergone already 6-7 lines of prior therapies. Usually they have not responded to the last therapy before they came into our study. And now we see this huge number of responses. It's unprecedented. And what's even more rewarding is that above 70% of these patients have a complete response.
Wow.
So now you can see what you can do by activating innate immunity. You can generate, if you find the right combinations, so in this case, the bispecific antibody, and natural killer cells derived, in our case, from cord blood. And this combination has a very high response rate. Initial data also on durability looks pretty good. And as I said, it's safe. So patients can be treated mostly outpatient. And after it's infused, they can walk home.
So at this show, there's been a ton of conversation that I've heard about cell and gene therapies, and I don't know. Stop me now if you don't consider yours to be in that category. But
Yes.
what I hear is there's two big challenges that are emerging, and they don't have anything to do with the efficacy of the drug. There's manufacturing, which turns out to be more complicated and more have more of an impact of efficacy than it does in small molecule.
Yeah.
And there's just the payment models. These are so effective that we don't know how to charge for them.
Mm-hmm.
So one of those might be a little premature for you guys where you are now, but how do you think about some of those challenges as in this new world of cell and gene?
Yeah, indeed, it is a challenge. Affimed's mostly a bispecific company, despite the fact that we're using a cellular product. So the manufacturing of our bispecifics has been optimized over the past years, and we can make large quantities at very reasonable costs of goods. The cellular product, so these cord blood-derived natural killer cells, indeed, is a very sophisticated manufacturing that initially do at a very small scale. So you can produce material for maybe 5-10 patients with a single manufacturing. So you're already seeing that that is not sufficient to supply the market ever. And costs are fairly high. So you talk about a 5-digit number.
Okay.
Still, this is what you do in the early days. And indeed, this technology was at that time when we started only available at MD Anderson. So they were one of the front runners to produce these natural killer cell products. It's allogeneic. So that's the beauty. And in the beginning, you don't worry because you have to first see, is it safe? Does it produce efficacy? Does it show initial good durability? And indeed, what happened to us is we've suddenly had this stunning data, and everybody was asking us, can you now take it forward and bring it to the market?
Right.
And here we said we couldn't do that because now to establish this manufacturing process could have taken us maybe 3, maybe 4 years. So it's a very lengthy process that you would have to invest in, and it's also expensive. At the same time, when we were looking into what we could do with the NK cells from MD Anderson, we obviously in parallel went out to check companies that are developing NK cells as a product. And we identified a company called Artiva here in the US. And they had already many years ago started to produce natural killer cells in large quantities. Indeed, their technology and the manufacturing comes from Korea, from Korean Green Cross. And they already had established this manufacturing back in 2018-19. So what's different now? The AB101 product from Artiva is produced in large quantities. We can produce thousands of doses with a single run, and the costs of goods are pretty low. They have published these costs of goods, so we're using roughly 1 billion cells, and this would cost $1,300. So you see, there are challenges, but we have learned that biotech can address challenges. Just remind us on who has basically brought all the innovation to solve basically and address COVID. It were biotech companies such as Moderna and BioNTech, and that's what we're here for. So we need to be innovative and need to address these challenges, and we believe you can address.
And what about the commercialization side? I mean, are these therapies? I know that the question is sort of, is it a one-time thing, and then how do you charge for that compared to an ongoing therapy that a cancer patient has to have until they, well, for the rest of their lives, basically?
So this technology is not a one-off thing. Okay? So allogeneic cells, as you know, this is not your own immune system. This is a component of the immune system from a healthy person, in this case, cord blood. So essentially, our body would reject it.
So it does have to get boosted.
Exactly. So what we do is initially you give a fairly mild, what's called lymphodepletion. So you reduce the capability of the patient's own immune system to reject these natural killer cells. You create space for about 2-3 weeks. So you can do 3 weekly injections of these natural killer cells. Later injections would not result in a reasonable amount in the body because then it would be rejected. At the same time, when the NK cell is given, we also infuse our bispecific antibody. So as such, this forms the complex, then basically travels to the tumor and eliminates tumors. And we have very impressive results because we learned that we can not only treat patients that have let's say, smaller amounts of disease. These were really patients with bulky disease. Some of these patients came out of hospice, so they were super sick. And I come back to this derailing. You can send these patients back home to their families. They're suddenly there, they feel good, and you can repeat these treatments so often. So we are giving, in the meantime, up to 4 cycles in these patients.
And it doesn't have the side effects of something like that.
It has no side effects, or very limited side effects, yes. So patients are, indeed, in the beginning, patients are very sick. Okay? If you suffer from cancer, you have just, it's called B symptoms. So you have a lot of symptoms. There could be fatigue, there could be high fevers. They leave after therapy, and this happens pretty fast. They can leave the suite where they're treated in pretty good shape.
Wow. So I've been asking everybody a version of the same question, based on what you've heard here and also just what you brought in with you. What do you think are the biggest pharma R&D trends to look for in 2023?
Yeah, that's a pretty general question. Okay, I'm not
Pick one you can talk about.
Exactly. Like. Yes. Obviously, yeah. I just want to make clear that there are several trends out there that I've just heard of, but I don't have the detailed knowledge. I think that what we have been talking about cellular therapies now since many years, and despite the fact that there are always some initial successes, we always have some low-hanging fruits where you can have a win. We're now in the phase of optimization. So usually that looks like as, yes, this was the one of success and it will never happen again, but you have to wait another 5-10 years, and then you really see the fruits coming through. So it takes a while until it's ripened. And the fact is, it's manufacturing, it's finding the right patient population, it's finding the right diseases. So I think from all diseases out there, one of the most important ones yet to address is cancer. And there are different approaches. So we also see the mRNA technology coming around big in the meantime. We have seen the benefit for the vaccinations. So obviously what they try to achieve is not too different from what we try to achieve. Eventually, you want to create an immune response in the patient that protects the patient in the longer run. So here, where all these technologies are meeting, and I can imagine that the trend in the future that you bring together bispecifics. Checkpoints are very important because they've become a mainstay in the meantime. Cellular therapies as they become cheaper and cheaper, but also the mRNA vaccination technologies. And then you have a really good service to the patient. As I said, it can take care of the tumor by itself, by its own body, and can live a normal life. And it's also the threat is taken away from the entire family, where you don't know, is the cancer coming back? Is my whatever beloved person dying of this disease? And we see it still happen far too often.
So all of these different threads that people are pulling at, eventually we're going to figure out how to, not necessarily, but sometimes how to combine them and use them together, but more often how to sort of figure out what's the right one for the right patient and have the best outcomes.
It becomes, so initially you have standard therapies, but usually when you are beyond your initial, maybe first line, second line, so after that, it becomes pretty much personalized to the patient. Again, the disease turns out to be far more difficult as it has either not responded or became refractory. Right. So there is definitely a personalization necessary. Why is that? I believe, and not only I, but many people believe, cancer is a disease of your own body. Okay? So the kind of mutations you generate, the impact the quality of your own immune system has. All these are factors that play together. So what do I mean by quality of immune system? We're protected by our immune system in a way that any day we're generating thousands of cancer cells. They're just floating around in the body, and the innate immune system takes care of it. So now imagine if a cancer is occurring, there is also a contribution by the deficiency of the immune system. And that's all what we have to address and find out. And if you can, then you can basically address the disease within the patient. So cancer is very, very personal and can only for a certain group. You have a general answer, but at the end you have to personalize it.
You mentioned treatments that, that there is an initial success, there's excitement, and then there's this period of figuring it out where you maybe don't see a lot of publications, you don't see a lot of approvals, and then we get into the point where it's actually sort of a viable thing, right? Someone mentioned a very similar thing at a conference I was at the other day regarding ADCs, which you mentioned is your first product, that they had sort of a really promising start and then a little bit of a trough of people couldn't quite make them work, that there were things in the manufacturing, et cetera, that weren't there. And now, well, I'm curious, how do you see that sector of the market shaping out this year and beyond?
So what you're saying is correct. ADCs are very successful in the meantime, exactly based on improvements over time. ADCs in general sound very simple. You have an antibody, you dock a chemotherapy to it, and then it travels to the cell, and then it destroys the cell. It isn't that simple, because what really needs to happen is that the cell has enough of the receptors. Then it needs to be internalized. Otherwise, it is not killing the cell. It's just like floating around in the body. So you have to really be able to, at each step, find the right solution. Now, once it's internalized, the chemo needs to be clipped off, and then and then it needs to destroy the cell. So you're always measuring in between the side effects and the efficacy, and that's usually what we see with ADCs. But that's a successful world, but it's the world that we want to further enhance. Not today by replacing antibody-drug conjugates, but they are considered as chemotherapies. Okay, and chemotherapies leaves a damage behind in your body in healthy places.
Even targeted ones.
Even targeted ones, yes. There's no such specificity that can only destroy a tumor cell. That targets are usually expressed also on healthy tissues. So target exclusivity is a major topic for antibody-drug conjugates. Interesting enough, the targeting we do, we can use promiscuous targets. We do not see side effects on healthy tissues with our technology. Our second program, which is targeting EGF receptor. EGF receptor is widely expressed in the human body, in lots of places, in the guts, all in healthy tissue. So if you come with a drug that targets EGF and make it an antibody-drug conjugate, people have tried, super toxic. Okay? So you cannot really make an EGF-targeted antibody-drug conjugate still today. With our innate immune system approach, we have just recently shown that we have achieved what's called a recommended phase 2 dose. So you demonstrate that your drug displays all the activity that you need, and we have not seen any side effects, even at dosing further up. So we have a good therapeutic window. We are seeing the effect that we contemplated, that the tumor is taking up innate cells and also T cells, adaptive cells, and we're now enhancing this efficacy. Here the focus is to bring it together with a checkpoint inhibitor. We have treated only a very small number of patients today, indeed only 4 patients, of which 3 completed the first course of therapy, but one of these patients has a partial response. This is a gastric cancer patient that has been treated now in the meantime with 4 prior therapies. So we're the 5th in line, has massive skin metastases, and these skin metastases were reduced in a drastic manner. So this is again a body of evidence when you find the right combinations. So we're using our innate cell engager in combination with a checkpoint in order then to activate innate and adaptive at its full-blown potential, and then cancer is reduced. So still early days. We're now treating patients at higher dose levels with AFM24, and if things go as planned, we can start a larger, what's called expansion study in this quarter that could and would allow us to treat at least 40 to 50 patients with either lung cancer, gastric cancer, pancreatic cancer, and hepatocellular cancer. So you see there are multiple opportunities. So that's where we are seeing ADCs coming at its limit, where we can use novel technologies, where we have to use novel technologies such as ours. But there are many good examples where ADCs most recently have made a very strong impact.
Well, thank you so much for talking to me, and I'm sure we could go on and on. I think we're coming up against the limit of how long we like our videos to be, but you are a great explainer of these things, and I really appreciate hearing about what you're up to.
Thank you so much. It's a great pleasure talking to you.